I'm going to start out by showing slides of a disease you've already seen. We have diseases in native plant communities, but generally speaking, you have to make an effort to find them. And when you do find diseases, typically they are not severe unless humans have done something to change the system. And one of the ways that we do that most commonly is through the introduction of exotic microorganisms. And if you heard Dave's talk earlier, you know the chestnut blight story, and what happened, as is commonly the case, is plants that look completely healthy were moved, and plants are commonly colonized by microorganisms that don't induce any symptoms, but when exposed to a non-co-evolved host, they may be quite damaging, and that's what happened in the case of the American chestnut, exposed to a fungus that was a benign colonizer of Japanese chestnut and other species, but was devastating to the American chestnut, and by some estimates resulted in the loss of about 4 billion trees. And it isn't possible to put a dollar figure on that, but obviously it was a disaster, and we think of this as the gold standard of introduced pathogens, and illustrates the magnitude of the loss that can result. The disease I'm going to talk about is pitch canker, and this was introduced to the West Coast, not from the other side of the ocean, but from the North American continent. And it was a problem, a well-recognized problem of plantation-grown pines in the southeastern United States. And what I'm going to describe is the pathways by which we believe it came to California, got distributed within the state, and the manner in which the native plant communities have responded to the introduction. We have good reason to think that the pathogen was transported to California with seed, and interestingly, it was well known that the pathogen was seed-borne, and the whitish material you can see here on the pine seed is growth of the pitch canker fungus, and here you can see it growing out on a selected medium. So it was well known to people studying the disease that the pathogen was seed-borne, and it was also known that pines that occur in California were susceptible to the disease caused by that fungus. But in spite of that, there were no regulations to prevent the movement of seed. And presumably that's because there was no interest group advocating for regulations of that kind. Nevertheless, the people who were importing the seed should have been concerned about it, because the consequence of planting infected seed is that a lot of the seedlings die. So here you can see that whitish material, that's growth of the fungus on the stem of the seedling. And here the seedling got up without an infection, but the fungus has grown from the seed coat down to the cotyledons, and that seedling is going to die. So that's the immediate consequence of planting infected seed, but another important consequence is that it allows the fungus to become established in the soil. So the colonies that are indicated here by the arrows are the pitch canker fungus. It's not something that we think of as adapted to survival in soil, but if you introduce enough inoculum, it will remain present for an extended period of time. So what can happen then is you plant another crop of seed in that soil, and those seedlings become infected. And some of them will die, but unfortunately, many of them will not. So the seedlings you see here all appear healthy, but some of them have infected roots. So although this is a fungus that kills pine tissue above ground, it can colonize roots to a considerable extent without causing any damage. So what you end up with then is cryptically infected seedlings. And those seedlings can be shipped, and at the time, and this is Monterey pine, in the mid-'80s, it's a very popular landscape tree, so that was an opportunity for it to be moved around, and it remains popular as a Christmas tree. So many infected trees were shipped from a nursery. Seedlings that appeared healthy were shipped to choose and cut Christmas tree farms. And what happened at those locations is some of the trees became symptomatic, and obviously those trees were not sold. But other trees were only beginning to show symptoms. They were maybe a little bit yellow, and with some green paint, they could look good enough. And of course, You only need the tree to look good for a short period of time to be a satisfactory Christmas tree. But then, Christmas is over. And people lose interest in the tree, so it ends up outside, and if left intact and unattended, it will be attractive to bark beetles. Bark beetles can establish galleries in the tree, and if that tree was infected, when the larvae move out and mature as adults, they can encounter spores of the pathogen, pick up those spores, and transfer them to landscape trees, including Monterey pine, which proved to be highly susceptible. And this was a tree that was known to be susceptible to pitch canker. People in the southeast who were doing host range studies inoculated lots of species, including Pinus radiata, and they rated it as one of the most susceptible trees. And that proved to be the case when the pathogen arrived here. So this is a tree with a moderate level of infection, and everywhere you see dead needles or naked branch tips, there is at least one infection by the pathogen. And those infections often are mediated by twig beetles, native insects that make small wounds to test the branch to see if it's suitable for breeding, and that wound can become infected. And the disease intensifies simply by repeated infection. Eventually, some of those trees die, and infections on the main stem can result in extensive streaming of resin. The firewood that is cut from those trees can contain the pathogen, and it can also be a breeding site for beetles. And unfortunately, although it probably seemed like a good idea at the time, land managers at state parks and state beaches decided that these trees which had died, and at the time they didn't know why they had died, should be cut up as firewood and given to campers. So that's how the pathogen got moved up the coast. The wood contained the fungus, local beetles breed in the wood, the young come out carrying the pathogen and visit nearby trees. So ultimately, the fungus got spread throughout coastal California, and the area shown here is where we find the disease, varying levels of intensity depending on the location. These are isolated sites, mostly Christmas tree farms, but some landscape plantings. And the fungus requires conditions, it needs moderate temperature with moisture available, and that means, fortunately for us, it's probably pretty much restricted to this part of the state. There are certainly areas in the Sierra Nevada that would be suitable during certain parts of the year, spring and fall, but not the continuous good conditions that we have on the coast. And as you go farther north, lots of susceptible species, but generally too cool when there's moisture present. One area where we see active development of pitch canker is at Point Reyes National Seashore. There was a fire in this part of the park in 1995, and Bishop Pine Forest regenerated in those areas, and actually the Bishop Pine Forest has expanded considerably in those areas. And unfortunately, pitch canker has become established there. So you can see here there are a few dying trees. This is a picture taken in 2007 by the park ecologist, and 2 years later you can see much more extensive mortality. And we think that part of the problem there is overstocking, that the trees are regenerating at a density much higher than what the water table can support, so the trees look great for the first 10 or 15 years, but eventually became water stressed, and that definitely aggravates pitch canker. The trees are more sensitive to the disease under water stress conditions. So we established plots at Point Reyes National Seashore in order to monitor development, but we didn't start until 2011, and just in that period of time, this shows the percentage of trees in our plots that are infected, and you can see we're up to 89% as of 2013. So that just means that trees have at least one infection. We can also look at the severity of disease, which is a function of how many infections those trees have, and that's what's shown here. And over that same period of time, we've gone from 28% of our maximum severity rating to 57%. So we're seeing a dramatic increase in disease in Bishop Pine, and we don't know what the course of that may be, but there is reason for hope, and that comes from our experience with the disease in Pinus radiata, the native population on the Monterey Peninsula. We've been monitoring the development of pitch canker there since 1996, and what you can see in this graph, so starting here is spring of 96, and this again is percent of maximum severity of the disease, and this is averaged over 41 plots that we have in the native forest. A steep increase in the early years, and that's right about here when we published the paper and projected the apocalypse, basically. But to our surprise, it leveled out. And since 1999, we really haven't seen any significant increase. There's been up and down. And the reason for that, there are two reasons. In part, the susceptible trees are gone. The very susceptible trees have died. But the more important part seems to be that some of the trees recover. And here's what one of those recovered trees look like. At this point right here, about 5 years earlier, there was an infected tip. And looking from beneath the canopy, here's what that looks like when the picture was taken. So it's dried up, broken off, and the subtending branches has taken over. So that reflects two things. One, the fact that the tree can contain the infection. It's always true. Individual infections don't progress very far. And the fact that no new infections occur. So that suggested that these trees now, although obviously having been susceptible in the past, were resistant to the disease. And we could test that hypothesis by inoculating the trees. A simple test, we make a small wound and deposit a suspension of spores, and then return later to see what extent of lesion development resulted. So here's a case of a tree that's resistant. That's where the wound was made. Very little discoloration, very little growth of the pathogen. Here we have an expansive lesion, all that discolored area killed by growth of the pathogen. So we did that test in a number of plots where we knew the disease history of the tree, and the results are shown here. This corresponds to the length of the lesion, and the broken line corresponds to an approximate indicator of resistance. So almost all those trees that had been severely diseased were clearly resistant. This was a control tree, one that was not around during the epidemic phase, demonstrating the efficacy of the inoculum. So we got results like this at 5 locations, showing very clearly that trees that had been susceptible were now resistant, and this is what we call systemic induced resistance. So the resistance is induced by the exposure to the pathogen, and systemic because it's active throughout the tree and not just in the part of the tree that had been originally infected. So my argument is that this is a really important phenomenon in perennials, and in fact I would argue that we probably would not have long-lived perennials were it not for systemic induced resistance, because exotic pathogens, although humans facilitate the process of moving them around, certainly that has occurred throughout the history of life on this planet. So long-lived perennials need some mechanism to survive the initial period to allow time for genetic adaptation to a new pathogen. And this is something that can happen provided there is genetic variation for susceptibility. And this is the case, at least for pitch canker, happily. This is the distribution of susceptibility we see in a naive population. So we collected seed from native trees, grew them up, and inoculated them when they were quite young. And this shows the number of trees in each category. So this is intermediate lesion length. These are shorter lesions, hence more resistant trees, longer lesions, more susceptible trees. So this is exactly the kind of distribution that you would expect to see for a quantitatively inherited trait like height in humans. Most are concentrated at the median. So the good news is that over time, natural selection should favor the more resistant genotypes. And that means that this should be a component of our response to this, and I would suggest any invasive pathogen, and that is to promote regeneration, to increase the opportunities for the plant species to generate new genetic combinations. And there is the potential, at least, that some of those will prove to be resistant. The other important thing, of course, is to avoid moving the pathogen, and pitch canker, like so many other examples, illustrates the hazards of moving plant material. Seed, the single most important way that we've moved this particular pathogen around the world, live plants, and also firewood. One should give careful consideration to movement of that kind of material, and anything that can be done to reduce the risk of moving pathogens with them will help protect our native plant communities. So I will end there and acknowledge some of the supporters of our research and members members of my lab who contributed to the work. They're drinking beer here, that's why they look happy.